And get started. I'm here to welcome you all here. My name is Tony Schmader. I'm the head of the Department of Psychology. I hope I get a chance to meet you at the reception if I haven't met you already. Thank you for joining us here for our annual Quinn Memorial Lecture. I'd like to begin by acknowledging that the land that we're gathered on is the traditional, ancestral, and unceded territory of the Musqueam people. And we're very grateful for the hospitality of the Musqueam Nation for our ability to live, learn, and work here on these lands. Here in BC, in the Lower Mainland, we're often also on the unceded territory of the Squamish, Tsleil-Waututh, and other Coast Salish peoples. And for those of you who are joining online, we have many people who are online as well. I invite you to take a moment to drop in the chat or recognize the lands where you are. It's my great pleasure to honor and celebrate the namesake of today's event, Dr. Michael Quinn. In the 1960s, Michael was an undergraduate student here at UBC. He got his BA here, followed by a master's degree, and in 1969, he became the second ever PhD student to receive a doctorate from our psychology department. He provided a generous gift to our department in 2006. I should mention he went on to have a distinguished career as a clinical psychologist at Riverview Hospital in Coquitlam. And his generous gift over just 2 decades ago today, this is the 20th year anniversary of the endowment that funds not only this annual memorial lecture, but also research and travel opportunities for undergraduate and graduate students in our department. It's a real pleasure to welcome 2 of Michael's family members here with us today. Agnes Douglas and Wendy Sutherland are here to join us and to celebrate the legacy of Uncle Mickey, as they would refer to him. And I'm really so delighted to have you here to join us both in this learning and this community building event that we've been able to have going on 2 decades now. So I'm going to next invite Dr. Daniela Palumbo up to the stage to invite today's speaker, Dr. Lila Davachi. And after the talk, we're going to have Q&A and then followed by a reception. So thank you again.
Good evening, everyone. Before I introduce our speaker, I'd like to mention that we'll be having a Q&A after the talk, as Tony mentioned, and we'll be taking questions from people both in person and online. To join the conversation, submit and vote on questions for the Q&A on slido.com using code #UBCQuinn. This year, we are so pleased to present Dr. Lila Davachi. Lila received her bachelor's degree in psychology from Barnard College and her MA and PhD in neurobiology from Yale University. She conducted her postdoctoral research at MIT in the Brain and Cognitive Sciences Department. Lila started her research group at New York University in 2004, where she was a professor of psychology and neuroscience and served as the director of the Center for Learning, Memory, and Emotion. She moved her research group to Columbia University in 2017, where she continues to conduct behavioral and neuroimaging investigations into how humans make memories. Lila's research contributions are widely recognized. She is the recipient of the prestigious Young Investigators Award from the Cognitive Neuroscience Society in 2009, and Columbia University's Lentfest Distinguished Faculty Award. She is an elected member of the Society of Experimental Psychologists and the Association for Psychological Sciences. Okay, when I try to think of how to describe Lila's work, one word comes to mind: transformative. Because if you look back over the last 20 to 25 years in our field, there were moments when we paused to ask ourselves, what should we be doing to advance our understanding? Which burning questions should we tackle or even anticipate? In these pivotal moments, there was Lila. With too many examples to name, I will highlight a few. Her neuroimaging work in memory has illuminated how the brain replays our lived experiences to transform them into enduring memory traces. Her work on event segmentation, or event boundaries, has shown how the brain parses ongoing experiences into discrete moments to scaffold and organize memory. To give you a sense of her influence, we just returned from the Cognitive Neuroscience Society meeting, and you would be hard-pressed not to find Lila's name in a poster session, symposia, or data blitz session. Just like event boundaries, Lila's work shakes things up, forcing us to reorient and change our old ways of thinking before settling into a new intellectual landscape with lessons to last a lifetime. Lila, we are thrilled you are here with us tonight. Please, everyone, join me in welcoming our distinguished speaker. Thank you.
I'm blushing. Thank you for that very kind introduction. It's really nice to be here. Just want to make sure I get my pointer ready. So with that introduction, I will just say that the lab has been really interested in one fundamental question, which is how our memories formed, and we've looked at that from the vantage point of how experiences, everyday experiences, get transformed into memories. But today what I'm going to talk about is what happens after an experience is over. So we've spent about 15 years trying to understand the post-encoding time period and how memories consolidate and how they might transform and change over time. So let's start with a simple schematic of the life of a single memory. And what I want to highlight is that a lot of work, my work, but everyone's work in the field, has focused on the encoding or the time of the experience and the retrieval. And if we think about it from the perspective of the brain, what we know is happening, or what we think is happening, there's always more to discover, is that at the time of an experience, there is a distributed pattern of activity. So each of these nodes might refer to activation in neurons in the brain. So there's a distributed pattern of activation that characterizes our experiences. And then later on, when given a partial cue, someone might ask you what you had for breakfast, or you might see a face on the street. It's thought that that original pattern that was present during the encoding, the experience, gets reinstated or pattern completed, allowing us to recover some of the details from that past. And there's a lot of work, more than is even listed here, that shows that the extent to which encoding neural patterns are reinstated during retrieval predicts memory success, whether you remember something. It predicts the vividness of memory. It predicts your confidence in memory in different neural circuits. So this has been a very kind of a powerful approach that neuroscientists of memory have taken. But I want to highlight that there's another time period, this so-called offline state. So the time between the experience and the retrieval, when you're not thinking about the memory. And that could happen immediately after an experience, where it's thought that, and we have evidence to show that those activation patterns will replay or reactivate in different neural circuits. So it's literally as if the brain is working for you when you're not working. And so that's what I want to talk to you about today, our attempts to start to look at memory reactivation during offline states. We were motivated by the large neuroscience literature in rodents, recording from single neurons in a brain structure called the hippocampus. What I'm showing you here is taken from kind of a foundational paper. If you have a rodent running on a maze, each of these pastel colors, it refers to a single neuron firing in a particular temporal order on the maze. And what was discovered is that when the animals were sleeping, neuroscientists could record those same neurons firing in the same order during sleep. Importantly, sleep replay, as it's referred to, is temporally compressed, so it happens on the order of 200 milliseconds, even if the experience unfolded over many, many seconds. And that temporal compression is really important to produce a long-term memory. It's really good for synaptic plasticity. Future work after the discovery of hippocampal replay had shown that if you're able to electrophysiologically suppress or interrupt replay in the rodent hippocampus, then the animals seem to forget or their spatial memory is impaired. So there's a little bit of a causal link that also emerged out of that literature. Now, I want to propose that reactivation is our current leading mechanism that supports memory strengthening and consolidation. As much as the encoding period is important and your ability to retrieve, there's lots of factors that can interfere or promote your ability to encode or retrieve, but reactivation is an independent time point, this offline state that can promote memory. So what's the evidence for that? Well, neurophysiological patients with hippocampal damage have anterograde amnesia and temporally graded retrograde amnesia. So if the hippocampus is either fully removed or impaired, patients won't form any new memories. So we know the hippocampus in particular is really important for our long-term memory formation. And although there is debate about whether detailed episodic or event memories ever become fully supported by extra hippocampal networks, now I'm talking about the cortex, it's clear that brain networks that are active and support memory retrieval do indeed change over time and become increasingly distributed across hippocampal networks. So I just took a big leap. What I'm trying to say here is that the hippocampus is really critical in the initial formation and reactivation of memories, but part of the job of reactivation, we believe, is to distribute those memories across cortical networks, sort of offloading the representation of those memories into more durable, resistant memory traces. And as I said earlier, the current leading mechanism thought to support memory consolidation or strengthening is memory trace distribution, as I just mentioned, and we think that involves repeated memory reactivation, as if the hippocampus is teaching the cortex what these memories are. And so we know that we expected when we started this work that we would measure reactivation in the hippocampus. But we also we're interested in looking at cortical networks to try to understand the co-activation of memories in the hippocampus and the cortex to understand how these memories become distributed. And so I wanted to highlight one now not so recent paper, but really motivating to us. Nikos Logothetis did a really incredible experiment where in non-human primates, he was recording from the hippocampus, measuring replay moments of reactivation. And what's graphed here in the red are parts of cortex that show an increase in activation contemporaneous with hippocampal replay. So the idea here is it's not just the story that the hippocampus is replaying these memories, which is quite incredible, but at the same time that those replay events are happening, the cortex is lighting up and activating. There are also parts of subcortex that are deactivating. So the idea here is there's an orchestra happening. It's not just a local replay event, but it's timed with what the cortex is doing. And a recent paper that was recording from patients who were awaiting neurosurgery. This is an opportunity to be able to record from single units or neurons in the human hippocampus, has shown that contemporaneous with hippocampal reactivation, each of these red dots refers to parts of the cortex that are showing increases in neural activity. It's actually measured high-frequency activity in the cortex. So it's not just happening in monkeys, but we know now that in human brains, this kind of orchestra is going on at the same time. So what I'm going to try to tell you about today, I have three parts to the talk. The first is I hope to convince you that in our work so far, we believe the most diagnostic biomarker of long-term memory is the distributed representation of an event across hippocampal cortical networks. So not just a local representation, but a distributed representation. And then we started to look at offline reactivation and hippocampal cortical connectivity, and it doesn't just reflect what you experienced. In fact, we think it's an active time period where the brain is selecting what to consolidate and what not to consolidate. It's not that we know this, it's not that we remember everything. In fact, we often forget most of what happens in the day, and we think that's part of how the memory system is designed. It's going to adaptively upregulate and consolidate information that's relevant to our goals. And then if we have time, I'm going to talk about how during offline states, memories are also reorganized. So instead of just strengthening memories, we've shown that the brain actively has the ability to reorganize memories according to their overlap, their shared features. And we think this allows you to build knowledge. So that memories in this model are not really for remembering, although we love to remember, but we really acquire and maintain memories to be able to update what we know about the world, to have more accurate models of the world. So let's start with this first point. I just wanted to talk about some of the methods that I'll use in this talk. We've gone about this research in many different ways, but the studies and the analysis that we do all have the same structure. So we give people an encoding task outlined here in blue, and many of these studies, if not all of them, have a baseline rest period. So this is our offline state. People close their eyes and they're just resting in the fMRI scanner, and then we also have a post-encoding baseline rest. And in all of these studies, we want to look for experience-dependent changes in brain activity or connectivity. In other words, how did this experience alter your brain networks? And we've done that in multiple ways. We look at local activation patterns. So we can look at, we can create a vector that represents activity in the hippocampus. This is a hippocampal ROI, a region of interest. And we can ask whether encoding patterns look similar to post-encoding patterns more so than they do to baseline patterns. In other words, that tells us that this pattern of activity is moving forward in time. I'm going to skip over B because I'm not going to show you any data, but we can look at multivoxel correlation structure. But what we're going to start with is looking at connectivity. So in other words, we can look at how correlated the hippocampus is with cortical regions in their time course during encoding, and then we can ask if that correlation persists into the future more so than it was evident in the baseline rest. And then once we have these measures, we correlate them with memory performance. So we can ask, how does persistence of brain activity relate to memory consolidation? So in the first study, this was conducted by Ariel Tambini when she was a graduate student in the lab. Again, we had people performing a task. This is called the object face task. People saw an object on a face on each trial, and they had to decide if the person was happy holding that object. We just want people to engage with the stimuli to form an association. And we had a baseline rest period for 8 minutes, and then a post-task rest period for 8 minutes. And what I'm going to show you here is a measure of correlated activity between the hippocampus and a visual cortical area called lateral occipital complex. This is a region that we know is really responsive to objects and faces, and we knew it would be activated during the task. And what you can see here is that connectivity between hippocampus and the lateral occipital complex was significantly enhanced after this experience with objects and faces. So this was the first evidence that hippocampal cortical connectivity was sensitive to your experience. So if you just, let's say you leave a symphony and you're listening to music, that when you leave that symphony, your brain continues to really reactivate the regions that were active during that experience. But does it relate to memory? So what I'm going to show you now are correlations between, on the x-axis is associative memory. So I should say that after this experiment, people were taken out of the scanner and we tested their memory for all of the objects and faces that they saw in the study. And they saw about 64 of them. So this was a tough memory task. So you can see associative memory on the x-axis as correlated with connectivity. This is before the experience, the baseline rest, and then after the experience. And you see that at baseline, there's no relationship between your resting connectivity, people coming right off the street. There's no correlation between the level of connectivity between the hippocampus and the lateral occipital complex and memory. But after 21 minutes of the object face task, now we see that the level of connectivity significantly predicts your ability to remember the objects and faces. So this was really important because we wanted to understand whether this experience-dependent plasticity in the brain was related to memory consolidation, and it looks like it is. Now, Ariel went on to her postdoc with Mark Desposito, and she ran the same exact study. Here's the schematic where people encoded objects and faces. There's a baseline rest, and then a memory test, immediate memory test. And after that test, she used TMS to causally interfere with activity in the lateral occipital complex in order to ask whether that connectivity was causally important. Did you need to have hippocampal LO connectivity to promote better memory? So they either applied the stimulation to the lateral occipital complex or to a control site. And what you can see here in memory is that when TMS was applied to the lateral occipital complex, remember, this is after learning. You're not interfering with the learning. You're interfering with activity during the rest period. That memory was significantly impaired for the information that was encoded, but it was not impaired when TMS was applied to a control site. And more importantly, she replicated in the control stimulation subjects. You could see there was a relationship between hippocampal LO connectivity and memory, just like she had shown in the earlier paper, but that relationship was eliminated. So now there was no relationship between connectivity and memory. So this was a really important step in moving beyond correlations to causality, to know that this post-encoding time period is actually really critical for memory consolidation. So what you do after an experience is equally important to what you do during that experience. So in this next study, we wanted to measure memory representations before and after they've had more time to consolidate. So we still want to understand how memories are distributed across hippocampal cortical networks. So in this study, what Kaya did when she was a postdoc in the lab is we had people study paired associates, just like the ones that you saw prior, on 2 different days. So on day 1, they studied one list. Then they came back the next day and they studied another list. And then we put them in the scanner, the fMRI scanner, and they reactivated paired associates or memories from long time ago, day 1, or just from 20 minutes ago on day 2. So we've given these memories either a day to consolidate and undergo reactivation or only about 20 minutes. Now what I'm going to show you is activity during the reactivation period in the brain. Importantly, she also tested memory at 2 time points to allow us to measure the rate of forgetting across time. In some of our studies, we look at memory only at 1 time point, but to really capture the consolidation of memories, the strengthening and the durability, it's typically better to look at 2 time points because you don't just want to know how much you remember, you want to know that those memories are resistant to forgetting over time. So I'm going to show you data from the hippocampus and another cortical region called the perirhinal cortex, also very sensitive to objects. Here is a little peek into the stimuli that we use. We used word-object pairs. We also had word-scene pairs to be able to look at the specificity of these relationships. And what I'm showing you here is the level of connectivity between the hippocampus and the perirhinal cortex as a function of whether the memories were encoded yesterday, the long delay, or just encoded prior to scanning, the short delay. And you could see that during the reactivation of these memories, there's greater connectivity in the long delay versus the short delay object memories. And we used these other scene word pairs as a control for this particular cortical region. So it's not that all cortical regions enhance connectivity, it's the ones that were engaged during the experience because it's a reactivation of that encoding activity. So the way that we think about this result is that when people are reactivating memories from a day ago, they already look more distributed, right? It looks like they're encoded not just in the hippocampal networks, but across the hippocampus and the cortex. So we're trying to track these over time. But does that relate to memory? We want to understand, is that distribution across the hippocampus and the cortex related to better memory? So what I'm showing you here on the y-axis here is the connectivity measures between the hippocampus over here and the perirhinal cortex. And these are the different conditions. The long delay is in red, the short delay is in blue, but we also had novel trials in the scanners. These are brand new paired associates they were exposed to. And what is on the x-axis is the amount of forgetting. So here, the further you go out, the more forgetting you have, which is worse. So you want less forgetting over time. And you could see that only in the long delay condition was there a relationship between connectivity and reduced forgetting. So the more distributed those memories had become over that 24-hour period, the less likely you were going to forget them over the next 24-hour period. So we think that's another piece of evidence that this is a mechanism of memory consolidation. And we did not see that connectivity related to these short delay memories, and it didn't relate to subsequent forgetting for new experiences. So we think that this connectivity develops over time, and it's a marker of consolidation. And just to check the data that we had collected, we looked at novel experience, we looked at activation, overall activation in the hippocampus during these novel experiences. Those predict subsequent memory for novel experiences, which the field had shown before. So when information is new, the amount of activity in the brain is a really good predictor of your subsequent memory, subsequent consolidation. But if those experiences are older and familiar, the connectivity is a better indicator of their subsequent consolidation. So really these different neural substrates exist, whether or not those memories have had a chance to consolidate. So there's other work that have shown related findings. So post-encoding hippocampal VTA connectivity. The VTA is a part of the brain that's very sensitive to reward. Connectivity with visual cortex is modulated by reward. And there's selectivity of post-encoding connectivity between these regions predicts different forms of later memory. And other people have shown that post-motor sequence learning connectivity in motor cortex predicts long-term memory performance improvements in the motor domain. And actually, we've also shown that hippocampal amygdala connectivity, again, the amygdala is a region that's really important in salience, is reinstated after emotional learning, and maybe we'll have a chance to talk about that later. This is making the point that not only can we measure the post-encoding persistence of connectivity, but actually it could influence how we encode new experiences. Okay. So let's move to the second part of the talk where we want to talk about, we kind of want to peer into the brain and understand what the brain is reactivating. What information from an experience does it choose to selectively reactivate and consolidate? And so this is a study conducted with Deepu Murthy, who was a postdoc in the lab, and he became really interested in a paradigm that's called motivated encoding tasks. So this is a task we tell people before they see a paired associate whether or not we're going to pay them a lot of money or a little bit of money if they remember that. This is a really good way to motivate humans. That's not surprising to anyone, okay? So the green star means if you remember the upcoming paired associate, you're going to get paid $1 later, and the gray star means you'll get paid something, but it's only going to be 10 cents. So you can modulate your sort of effort to encode. And they do a bunch of these trials. So if they really remember these trials, we actually did pay them the amount of money that they earned. So we didn't lie to them. And what you can see here is that reward-motivated encoding actually works. So people are better able to, they remember more of the paired associates in the high reward condition than in the low reward condition. So long-term memory is significantly enhanced for the high reward-motivated information. But what we wanted to understand was what was happening before the memory test during this post-encoding offline period. So here I'm going to show you analyses targeted to hippocampus. We looked both in the anterior and the posterior hippocampus because prior work had suggested that anterior hippocampus is more innervated by the neuromodulator dopamine, which is really important in reward-motivated behavior. And then in order to look at visual cortex, we highlighted regions of the visual cortex that were in the high reward condition or the low reward. So here for faces, we look in a region of the visual cortex called the FFA, and for scenes, we look in a region of the cortex called the PPA because they like to respond to these stimuli. But importantly, we controlled what region we were looking at, and I'm just going to refer to the visual cortex as category selective cortex. Whether or not you were cued, the high reward was always faces or the high reward was always scenes. I should have mentioned that. So you either were rewarded for remembering faces or scenes, and then we counterbalance that across participants. And one of the findings that we saw was that what I'm showing you here is the change in connectivity between the hippocampus and visual cortex as a function of later memory. And we saw that there was a relationship between post-encoding connectivity and later memory only in the high reward condition. It was not significant in the low reward. So unlike the other studies that I've shown you where we didn't include any prioritization based on reward or emotion, people weren't asked to prioritize information. Here you're asked actively to prioritize. And now you see that post-encoding connectivity in the prioritized circuit is predicting memory for specific information that you encoded under the reward motivation. We did see that other parts of hippocampal cortical circuits were active and predicted memory for the low reward, and that was in the posterior hippocampus for those of you that are interested. So there's an interesting dissociation along the hippocampus. We also, so then we moved to asking, well, let me take a pause here. So now I think I've established that as soon as an experience is over, the brain is actively working to reactivate those experiences. So we've looked at that immediately after post-encoding. We waited a day and showed that its distribution over time is more predictive of memory. So in this study, we wanted to ask, can we accelerate consolidation? In other words, in the context of this work, can we accelerate the distribution of memories to the cortex? Because that seems to be what makes them really resistant to forgetting. And so what we did is we simply had people restudy the information during the task. So far, everything I've shown you has been only seeing a paired associate once. So let me take you through this design. This was done by Wangjing Yu and Asia Zadbu, two of my trainees from the last few years. Okay, so in this study, again, we're showing people paired associates. Here it's a word scene or a word object pair. And there's a baseline rest and a post-encoding rest. We had two versions of these, so we could look at post-encoding twice. We just kind of broke up the learning because there was a lot of information for people to learn. And then after that, we gave people a memory test where we showed them the word and they had to remember what object was paired with the word during encoding. Now, what we did in this study that was different is some of the paired associates were studied 3 times, where others were studied only once. Now, this should be familiar to all of you who study for an exam, right? If you repeat your study, you're going to be better able to remember that information. Like repeated encoding is really important. Not surprisingly, our participants had much better memory for what we call the strong paired associates, the ones that were studied 3 times compared to the weak memories, the ones that were only studied 1 time. Now, what I'm showing you here is measures of reactivation locally in the hippocampus for these 2 memory types. One of our hypotheses is that maybe the restudying of information should improve the frequency of reactivation events in the hippocampus, but we didn't see that. We actually saw that both weak memories and strong memories were equally reactivated in the hippocampus after learning during these baseline rest periods. What was truly remarkable is what we saw in the cortex. So these are different cortical ROIs, and I won't go into a lot of the detail except to say the MPFC is one that we'll focus on in subsequent slides. And what you can see here is that the frequency of replay events in the cortex was significantly enhanced across all cortical ROIs. And we did a follow-up exploratory analysis of the whole brain and saw that this was evident in many of the cortical regions that we looked at. So simply repeating or studying paired associates 3 times led to what we think of as an acceleration or movement of the distribution of those memories to cortical systems. It's not that those memories aren't represented in the hippocampus. In fact, we think there still are, but the weight of that reactivation is now pushed to your cortical networks, which are more stable and will allow those memories to be around longer. We also looked at the number of co-memory activations. So in this analysis, we're asking how often is a memory replayed in the hippocampus and the cortex at the very same time? Our resolution here is on the order of a couple of seconds. So we can actually try to understand whether the hippocampus is teaching the cortex. Is it actually spreading out, these memories to the cortex? And we saw that only in the medial PFC. So in the hippocampus and the medial PFC, a region that is really important in the representation of consolidated memories, shows greater coordinated replay events after strong learning compared to weak. So it looks like this kind of simple manipulation of restudying is actually improving the hippocampal cortical dialogue. It's improving their ability to speak to each other and represent these memories. Well, what about behavior? Does this relate to behavior? So what we found is that in the hippocampus, when weak memories in green were replayed, this rescued them. There you go. Now we're all going to remember this moment better. So it's even though the overall frequency of replay events were similar for weak and strong events, those weak memories seem to really rely on the hippocampus to rescue them. So the more frequently a weak memory was replayed correlates with your ability to later remember it. So it really predicted it. But it didn't do so for the strong memories. Instead, what we found is that a cortical region in the prefrontal cortex, so the extent of replay of information of strong memories as well as weak memories, predicted memory in the cortex. So here, what this is telling us is that hippocampal replay is happening equally often for weak and strong memories, but it's really important for weak memories. In other words, in order to lift it up, to be able to spread it out to cortex, you have to really replay those memories. Cortical replay is happening more for the strong memories, and it also predicts your ability to remember that information. So again, it's another piece of evidence that consolidation is kind of distributing these memories across these networks. Okay, so I hope I've convinced you that memory activation in the hippocampus and hippocampal cortical connectivity is really important for memory consolidation and strengthening of those memories. But we also know, as I noted at the beginning, that probably the strongest rule of memory is forgetting. So why would a memory system be built and then there's a lot of forgetting? Well, inherent in just that observation is that maybe the memory system didn't develop for us to remember everything. Maybe it developed for us to be able to take the pieces of our daily experience that are surprising and new and relevant and to be able to update our models of the world. So we started to develop studies that allowed us to understand if the hippocampal and cortical replay mechanisms are sensitive to the structure of a learning experience. So let me explain that to you when I show you the next experiment. Hopefully it'll be clear. So I'll give you an example from my everyday life. I love coffee, and the first time I went to a coffee shop called Blue Bottle, I don't know if you guys have Blue Bottle here in Canada yet. Okay, so it was the best coffee I'd had at the time. This is in San Francisco, so I formed a very distinctive memory of that Blue Bottle experience in the Ferry Building in San Francisco. I believe this original Blue Bottle is still there. Some number of years later at a conference in Chicago, I found out there was a Blue Bottle, so I walked the 12 minutes to go get my cup of coffee at Blue Bottle. And again, I formed a distinctive memory of that experience. Now, what's interesting is that after now I have a Blue Bottle a block away from my house, so you can imagine how happy I was when that happened, although it's very expensive, so I don't go there as often as I would like. After many repeated experiences with very similar features, so Blue Bottle has you have an expectation of what it's going to look like with the light wood and the blue bottle, and it's very clean, and the aesthetic is very similar across those stores. So over time, you can develop a representation of what I call blue bottleness, right? So these sort of orange features, you have a sense of blue bottleness. And this is really part of what our knowledge is, our semantic knowledge. Even if I forget the individual episodes of visiting Blue Bottle, which they're slowly fading, I still can tell you what it means to be in a Blue Bottle cafe because I have that representation. So what we wanted to understand with this next study is instead of presenting people with paired associates that have nothing in common except maybe time with each other, we presented paired associates, we asked people to learn the individual paired associates, but they had something in common. So for each trial, people saw an object and a scene, but there were only 4 scenes across the whole experiment that repeated. The objects were always trial unique. So the objects were always novel, but as you can see here, we presented beach scenes, actually only one beach scene, a jungle scene, a city scene, and a bedroom scene, and those repeated 28 times. So you're learning something new along with something that's repeating, which I think mimics a lot of what our daily experience entails. And what we wanted to understand is, does the cortex, in particular, we were interested in medial PFC as well as the hippocampus, come to represent those experiences? This isn't working how it's supposed to, sorry, along their shared features. So will the jungle memories all come to be represented in overlapping neural networks in the cortex? Is this our way of representing knowledge? Is it that they're represented in overlapping networks? And as distinct from all the bedroom memories and all the beach memories etc. So we wanted to know whether the brain, whether and how the brain starts to organize our individual experiences. And so this was conducted by a grad student in the lab at the time, Alexa Tampari, and I'll just give you some details about the design. So here, during day one, people encoded 128 pairs, and they were studied 3 times. We wanted to make sure they were remembered at a week delay, and I'll tell you why we did a delay. And then we had an immediate retrieval where we expected memories to be still separated. We knew people would still have access to the individual experiences. And so we tested half the memories right away, but we waited a week and brought them back and tested their memory on the other half. So this week period allowed time for those memories to reactivate and to be distributed across the cortex, and hopefully enough time to reorganize if that was going to happen in this week period. We really had no idea of the time scale of what we should try to do, but we also knew that we wanted to make sure they remembered the paired associates. So we tried for a week, and it looked like it worked. So here is an example of a particular trial. They would see an object and a scene, and then they were asked how vividly they could imagine these 2 things together. We always ask people a question to make sure they're awake in the scanner and they're pressing a button. We want to make sure they're doing the study. Importantly, during retrieval, we no longer showed them the scene stimuli. The scene stimuli are the ones that are repeating, and that's our feature that links them together. So now they're only looking at objects, and they're in the scanner while they're retrieving. And so we show them, in this case, these flippers, and we ask them what scene was it paired with. So to tell us which of the 4 scenes it was paired with. And we did that both at recent retrieval and at remote retrieval 1 week later. And not surprisingly, we see that people's ability to remember the scene is much better right away. They have better memory, but they're still above chance at 1 week. So we had enough trials to be able to analyze the correctly retrieved paired associates at 1 week. So how did we analyze this? In this experiment, what we did is here are the individual trials during encoding. Now they're organized by the shared features. So these are all the jungle memories, and then here are a couple of the other memories. And what we can do during retrieval, when they're only looking at objects, is we can look at whether there's similarity in the retrieval neural patterns for objects that were learned with the same scene. And we can see if they're more similar to each other than they are to other objects that were learned with different scenes. So it's our way of understanding whether all the jungle memories are clustering together in the same neural ensembles, and they're represented distinctly from the other scene memories. And what we found, I'm going to show you data from the medial PFC from this region of interest. What I'm showing you here is the inter-item similarity, how similar trial neural patterns were to each other when they shared a scene, that's the overlapping, than when they didn't share the same feature, the non-overlapping. And you could see that at recent retrieval, there's no difference in their overlap. It looks like they're represented just as they are, and there's no clustering of information. But at the 1 week time period, what we see is the emergence of structure. So now this cortical region is representing information as the brain has made sense of it, as I presented to you. So jungle scenes are represented in overlapping neural networks and distinct from other scenes, as if they're now clustered into these different neural kind of neighborhoods in the medial PFC. We also saw that organization in the hippocampus, which was very surprising to the field because the hippocampus is particularly important from prior work in representing and consolidating episodic memories, but we've shown that it also was really important in organizing that information. So in this whole hippocampal cortical network, you see with a week's delay that now the brain is not just representing the episodic information, but instead it's prioritizing the overlapping, the semantic structure of that information. And that may be why we remember the gist of our experiences a lot better. You might intuit this from your own world than we can remember all the details from our past. We then asked if this neural overlap that we see or clustering at 1 week is related to immediate post-encoding connectivity. We wanted to know, does the brain immediately after those experiences start to take advantage and start to teach? Does the hippocampus start to teach the cortex? And so what I'm showing you here is hippocampal MPFC connectivity on the x-axis as a predictor of retrieval similarity in the hippocampus, that data point that I just showed you. So how much does your immediate connectivity relate to clustering? And in the recent time point, there's no relationship. But at the 1 week time point, we now see a significant relationship. So your brain's not waiting to reorganize these memories. Immediately after an experience, the extent of connectivity or communication between the hippocampus and cortex predicts the extent to which your brain will be clustering that information at 1 week. And then we moved on to other kinds of regularities. So beyond featural regularities, we moved to sequential regularities or time. So in this study, people are in a continuous object recognition task. Now they're just seeing objects. And what we've organized the information so that object A always is followed by object B 100% of the time. So you can build up a prediction that you're going to see the broccoli after this tool. I can't see what that is on my screen. Oh, it's a can opener. Okay. But the B object has a 50% transition probability to these two other objects. In other words, A and B will predict that you will end up at C1 or C2, but C1 and C2 are never seen together, but they could be temporally linked through their preceding context. And we were really interested in whether the brain would start to organize C1 and C2 as if they're related to each other. So to put this into maybe into more of your daily life, if you always are either leaving your home and going to work or to the grocery store, but you never do those things in order. They're never connected. You either are doing one. Do you come to now represent those two destinations in a similar way? That was the question we wanted to ask. I'm going to skip over the learning. They learn these transition probabilities. And what we did here, in order to test the extent to which these are overlapping, the C1 and C2, we did a priming task where at our 24-hour delay, we just asked people to simply say whether each object was old or new. And then we ordered them so that the C1 and C2 objects were presented next to each other. And if we reasoned that if they're represented in overlapping neural ensembles, you should be faster to respond to C2 after C1. In other words, it primes your access to that information. And what is maybe hard to see here, but we did a behavioral experiment and then the follow-up fMRI experiment. I want you to focus on this remote time point. What you could see is that people were significantly faster or more primed to respond to C2 given C1, and you did not see that when we tested memory immediately. So the priming effect grew with time, and I would argue that we needed consolidation for that to happen. So we have a significant evidence behaviorally that they are now maybe represented in overlapping neural networks, but then we wanted to look at that. So in this experiment, we focused on MPFC, given our prior work, and created two different models that could look for whether this is the kind of pattern we're seeing. So let me take you through this. So let's just focus on the remote time point. This just refers to our ability to ask whether C1 items are more overlapping in their neural patterns with C2 items. And we expect that on the diagonal. Diagonal are C1 and C2 items that were preceded by the same AB context. So their temporal information is the same compared to all C1 and C2 items whose temporal information was not the same. This is just a way of analyzing the data to look at specificity. So is this particular C1 and C2 closer to each other than all C1s and C2s are to each other because of their same positions? And actually, we saw something that we didn't expect. So overall, in the MPFC, what we saw is that there was more dissimilarity between these two different destinations. So if you either go to home, or you either go to work, or you go to the store, it looks like the MPFC is separating those destinations out because they never co-occur. But the extent to which they are still overlapping predicted our priming measure, which is what we had hypothesized. So even though the main effect is to pull apart these two separate destinations, the extent to which they are overlapping predicts your ability to, your priming measure. So the extent to which you get faster when you see C2. So we kind of saw what we thought we would see, but also maybe something that makes a lot of sense. Like your brain is separating out these two destinations instead of integrating them as a main effect. And then finally, I know I'm getting close to being time, so maybe we wanted to ask, after this kind of learning, where you have 100% transition probability and then 50% transition probability, what does the memory system choose to replay during rest? So before doing this retrieval task, what does it choose to replay? And this is an analysis that was conducted by Arlene Lormistoir, who's currently a graduate student in the lab. So we had a rest period after this learning and before any kind of memory retrieval test. And just to give you the bottom line, the hippocampus is more likely to replay these B items and these C items compared to the A item. The A is the most predictive and the most reliable marker of these transitions, but it looks like the hippocampus is replaying the uncertain transitions, both the B and the C items more so than the A items. We also see that effect in the MPFC, so you get more frequency of replay of B and C items. And importantly, the extent to which B items are replayed in the MPFC predicts C priming. So in other words, the more the brain seems to, again, this is kind of a weak learning to like align it with that earlier study, people are less likely to learn these transitions than they are to learn the A to B. So it looks like the hippocampus and the cortex are taking advantage of this post-encoding time period to replay these uncertain transitions, and that helps them to integrate their outcomes. So replay prioritizes time points with higher uncertainty, and this is active work. This is the most recent experiment that we've conducted. I wanted to give you a sense of where we started and kind of where we're ending and the kinds of questions we're asking. Okay, so in conclusion, I hope I've convinced you that the distribution of a memory trace across hippocampal cortical networks is the single best predictor so far of memory stability, with a special emphasis on midline cortical regions, in particular the medial PFC emerges in all of our studies. And those also representing memory content, the visual cortex. Now, memory transformations from encoding to retrieval are likely becoming the rule for the neural representations of memories, not the exception. So memories are changing. I didn't talk a lot about that, but replay isn't just veridical. As memories are replayed, information drops out and other information grows, especially information that's similar to other experiences. And I think it's a really exciting time for empirical research because we're able to modulate the learning experiences of participants and then directly examine the features of those experiences that increase post-encoding reactivation. So it's like we create different worlds for the hippocampus and the cortex, and then we kind of listen and we're able to look at what the brain cares about to learn more about what it's sensitive to. We're able to probe what are the behavioral consequences, more importantly, of the underlying neural representations with an extensive array, I think, of really elegant assessments of behavior. We can look at memory, integration, we can look at the mistakes you make, if that is predicted by overlap, priming measures. And also, this is a prime target for manipulation and clinical intervention. So there are methods to increase hippocampal cortical connectivity that have been out there in the literature. And so I think this, as a biomarker of memory, is really exciting because we can modulate hippocampal cortical activity, not just in the lab, but also in the clinic. So I think that is a really good step forward. And I just want to thank all my students and trainees who did all the hard work. I get to be here. I'm lucky to tell you all about it. And all my funders, and thank you for your attention today.
Thank you for that. Testing. Oh, good. Thank you for such a wonderful and thought-provoking talk. Now we're going to open it up for questions. We can take questions in person, and then we'll be monitoring online for questions as well. So just raise your hand if you have a question, and we'll bring you a mic.
Thank you so much for that talk. I know we were focused on the basic science, but I was very curious to know a little bit more about what you said at the end about potential clinical modulation of connectivity, and also just any other implications that you would take from your work about, how to improve memory.
That's a great question. So the work that I was referring to was first done not in a clinical population, in a healthy population, where if you use TMS, which is relatively safe, non-invasive, it's basically you use a magnetic field, you can stimulate above the cortex, and this is work from Joel Voss. What he found is that if you stimulate repeatedly in sort of like in rhythm with a theta oscillation, there's reasons for that, If you find a target on the cortex that is already highly connected to the hippocampus, and you repeatedly stimulate that target over 3 or 4 days, they found that you could increase hippocampal cortical connectivity with that target, and more importantly, they improved memory. So their first paper was published in Science. They've replicated it several times, and his trainees are replicating it. So we know that it does take a little bit of work. You've got to come into the lab 3 or 4 days, but I think it's probably worth it. Now, they've only shown it so far in these very experimental designs with paired associates. And so a big question is, how do we take that and improve memory for real world experiences? And that hasn't been done yet. But I think now that we know we can modulate connectivity, we have to figure out how long that can last, and will it then, moving forward, improve your ability to encode and consolidate experiences. So it's relatively simple, relatively safe, and seems to be working, and it's reproducible. So we're really at an exciting time with that method. Other ways to improve memory is really fun, Daniela. You should, yeah. So we know that like emotion, reward, attention, novelty, one of the things we've been doing that I didn't talk to you about today is starting to do a little bit more work in autobiographical memory. And our first study asked participants to keep a daily diary. It really is just their phone. And we asked them at the end of the day to tell us about 3 events that happened that day, and tell us whether it was novel, whether it was routine or periodic, and then they describe what they did that day. So participants did that every day for 14 days, and then we waited 2 weeks and we tested their memory for those experiences. And what we found are that novel experiences are remembered with more episodic detail and greater vividness at this 2-week to 1 month time point. But even more surprisingly, what we found was that on a day where you had a novel experience, that the routine experiences on that day were also better remembered than routine experiences on a day without novelty. So we're learning that novelty in your everyday life, even though, interestingly, we looked at emotion, people may be a little wary, novel experiences were associated with a little bit more fear, a little bit more trepidation, but also related to greater mood at the end of the day. So it's sort of, this picture is like, it may be a little hard to get out of your everyday experience and do something new, but it's going to pay dividends both for your mood and your memory. So that's something that is really kind of a simple everyday thing, and maybe every other day, that you can engage in, is trying to give yourself some novelty. And we also asked people whether it was social novelty or spatial novelty. We measured how many steps they walked. It's a really rich data set, but it turns out that both social and spatial novelty are really good for memory and for rescuing those sort of weak experiences around that. So, yeah.
Hi, Lila. Can you hear me? Really interesting talk, and I have a question about the Blue Bottle Cafe, actually. So when you started talking about it, in my mind, I immediately started forming an image of me having been in the Blue Bottle Cafe. And my image was actually of having been in Vancouver. It wasn't that, so the image was, it must have been either at the ferry terminal of Tsawwassen, which is one of the ferry terminals around here, or another one which we have in downtown. And then you said, do you have Blue Bottle Cafe here? And I started nodding because it was so vivid in my mind that I was certain that we have it. But then you said, when I went to it, it was in San Francisco. And that reminded me that actually, when I went to it, it was in San Francisco too. Yeah. So I realized I formed a very, very vivid and high certainty kind of confabulation in my mind, which was dispelled by your account of, as soon as you mentioned San Francisco, I realized, yes, that's when I went to it as well. Of course, the CNS conference is going on right now, just finished. And that's actually when I've been to it. So my question for you is, when I first formed that confabulation that had a very high degree of certainty, enough for me to start nodding in response to your question, was I using more of my cortical regions to form more of a weak memory versus when later on you scaffolded my more correct experience, was I then descending further down into the hippocampal involvement in order to form the stronger memory of actually having been to San Francisco and going to that coffee shop?
I think that I'm glad that you asked this because it's important to highlight that at the time of retrieval, like I gave you a cue and your brain reactivated a memory. And what likely happened is exactly what you're saying, that what you reactivated first, these things are happening in parallel. You could have them both at the same time, but you might be reading out your cortical pattern, that there is the gist of being at Blue Bottle was very clear to you, but you got some of that stuff wrong, right? So your brain had kind of mixed up location. We might do this with all coffee shops. There might be like overlapping representations in coffee shops. We don't know all the features that are organizing our knowledge. We do know about categories that children learn and that we learn. We know that faces and scenes are separate and tools and animals and some of those features, but really in our everyday experience, we're not totally clear about how the brain is organizing that information. So I think what we would have seen if you were in a scanner is that you had cortical activation that then needed to be refined with additional details. And you may have gotten there without me, but you may not have, right? So the input that you get is like a key, right? It's sort of a key that can unlock the actual specific memory or not. And then hopefully going forward, you just formed a new memory too, which is this experience of having made that confabulation, and that you can use later on, right? So yeah, it's a good question.
Thank you so much. This was fascinating. I'm a clinical counselor, so my interest lies in how your research might inform trauma and memories associated with trauma. So especially when, like, this event, or I guess the offline time that you're talking about, like the replay, does that have anything to do with?
Rumination.
Yeah.
Yes. I think that is not something that I've studied yet. We've looked at, I'm doing some work right now at the very beginnings with social anxiety. It's not quite a trauma memory, but I expect the same thing to be there. So this is there's a healthy version of reactivation and replay, and then there's sort of an unhealthy version. People think of PTSD or maybe other kinds of trauma where either those experiences, I would say and, not either, but those experiences are burned in more tightly because of the neuromodulation and the heart racing and like a highly emotional experience, which is also correlated with better memory, but it likely leads not only to greater replay frequency, so you're really going to like stamp that memory into the cortex, but it might also create I think of it as like a deeper path. Like when you replay information a lot with a lot of detail, it might be hard to veer away from that particular neural pattern reinstatement. So there might be some memories that become so replayed that any little reminder, anything that at all overlaps with that takes you right back to that spot. And I think in a lot of, you know this better than I do, but from my reading of it, a lot of trauma memories have the same kind of start and middle and end. They're really repeated in exquisite detail, which is very different than our more healthy, kind of boring, neutral memories. So I think that's an example of this process gone awry. And it's really hard, as you know, to try to change that, to try to overwrite or make those memories less emotional, which I think a lot of the work now is trying to disentangle the physiological memory, the heart racing and all that from the content, because we're never going to get rid of the content. I think it's going to be really hard to have trauma memories be forgotten in their content, but maybe we can separate out the content from the upsetting nature of replaying that information. But yeah.
I have a microphone, so I guess I will speak.
Where are you? Oh, okay, there you are.
Thank you for your talk. I found it really interesting. So I'm lucky to do a bit of memory research myself, and I understand how there's like a big emphasis in the field on accurate recall and kind of durable, stable memories, and obviously that plays a big role in the research you presented here. But as you touched on at the end, that's not necessarily like an inherently maybe universally desirable thing. There are some theories out there that suggest that maybe it might be adaptive in some cases for memories to be a bit flexible, and you talked about how during replay, some details may drop out or maybe be added. I'm curious whether you think there's any role of these like hippocampal, cortical connections in kind of not just accurate recall, but kind of the adaptive use of memories and kind of almost flexibly thinking about memories or kind of playing with these memories and thinking about them in different ways that might enhance their use in future behaviors.
Yeah, I think that there used to be, I think, the lore, the theory about memory consolidation. I've only talked about it at like an hour, and then I've gone to a week. But the idea from the literature that was derived from studies of patients with hippocampal damage or rodents with hippocampal lesions is that after enough time, let's say weeks or months or years, that memories become represented only in cortical networks and not with the hippocampus. I don't think we have great evidence for that. I think that the bulk of the evidence now is that when you're retrieving even a very old memory, the hippocampus is still involved in whether it's the retrieval of that memory or whether it's in creating plasticity around that retrieval process. So we now know that every time you retrieve a memory, you open up a window. It's called a reconsolidation window. So it's not that it's consolidated and it's a stable thing. It's not an object in the brain. Every time you reactivate it, you can change it. It can be updated. And even your friends can change it. There's like social reconsolidation. If they say, oh no, Kalina, I was there with you at the blue bottle, right? So I do think that these windows of plasticity open up and the hippocampus is probably more involved in the long term. I don't have great evidence for that. We haven't done years-long memory and shown that that's the case, but I do think the work on adaptive memory suggests that this is primarily from rodents, but that memories are always being sort of changing, and that when we have these older memories that we recall in the very same way, in the same order, those are just the really embedded ones in the cortex that probably aren't benefiting from as much plasticity. But yeah, it's a good question. Thank you.
I'm going to take a question from online. So lots of great questions, and some of them were covered in the questions that were asked in person. There's one question that's been upvoted 6 times, so I'll start there. Why aren't dreams just a literal replay of waking experiences if the function of offline replay is to consolidate memories?
That's a great question. So I don't do sleep research. The initial replay events that I talked to you about in the rodent hippocampus was during sleep. Now, they're primarily seen not during REM sleep when you're dreaming. So consolidation processes in the brain are happening during non-REM sleep, so the opposite time. And that's a time where your brain, the hippocampus and the cortex, are actually in this nice orchestrated activity with each other. So they're communicating via these what's called slow oscillations, and it's this very beautiful non-REM sleep. REM sleep is when you dream. So it's really tempting to think that your brain is replaying experiences and you consciously experience them during sleep. Like that becomes a part of your sleep structure. And no one really understands, I would say no one really understands dreams, although someone's going to argue with me. But what I will say is it's happening at a different time period. We have really good evidence that non-REM sleep, slow oscillations during non-REM sleep are critical for memory consolidation. What I've shown you is that awake rest also is really important, those moments of replay, and that they might be related. So one line of work that scientists have been talking about, my colleagues have been talking about, is how does this awake reactivation tag information for sleep-dependent replay? So is it this, why would we have 2 time periods where the brain is replaying information? One when you're awake in your daytime, like you walk out the door, your brain is going to reactivate, and then we have this sleep. There's got to be a relationship between the two of them, and what are they doing differently? That's a really open question. But I can't tell you why we dream about what we dream about, and why it makes no sense, usually.
I think that was the conclusion of a sleep replay workshop that some of us went to a few days ago. Nobody understands dreams.
Yeah.
Okay, let's see if there are some other questions. Okay, yes.
Hello, thank you for your talk. I had a question about replay, and I'm curious if you have thoughts about what triggers this kind of spontaneous replay. And I'm interested especially in like how it comes up in life. I've noticed it arising in my own life. You're kind of going through something and it appears. And I also know it's talked about in different traditions, like in the yogic tradition, they talk about smriti is memory, and it's one of the afflictions that's because it's kind of always interfering with being present. So I'm curious if you have an idea of what is triggering these replays to happen, why specific things are being replayed, and what might be beneficial about this versus problematic. Like, are there kinds of replays that are like helping to strengthen the memory and useful versus like maybe if it's a negative memory, it's more problematic?
Yeah, absolutely. So what triggers replay events? Well, I think it's an endogenous biological process that has its own time course. One, like there definitely is. So this is why we just like listen in on the brain and we try to measure when we think certain memories are being replayed. That said, I do think there are brain states that can stimulate replay, especially hippocampal replay. And I believe that those are moments of high arousal, and they don't have to be emotionally arousing. So like in another line of work that Daniela mentioned, we studied event boundaries, and those are moments of transition and change between contexts. So think of this as one context, and then when you walk out the door, that's another context. And that's really beneficial for memory encoding because you have two contexts to like situate your memories in. But I think that the moment you walk out the door is actually a moment of high arousal. So we can measure your pupils will dilate, your locus coeruleus will activate and release norepinephrine. And I think that that moment of arousal, it can be emotional, it might stimulate replay just in terms of this biological system. So there might be moments that are particularly important for tagging memories. So if something emotional happens, the brain might have a mechanism to ensure that that memory is going to get replayed during sleep by doing a little mini replay. It's like a rehearsal. I don't have any evidence for that yet. I have evidence for the pupil dilation part, but not the reactivation. Oh, that's not true. I have one study, but I'm not confident. And absolutely, like I think, you know, adaptive and maladaptive is such an interesting question, and we only know that post hoc, right? Like it's so, I think that the structure of the world, any way in which things can be prioritized for you, and we started with simple things like emotion, reward, the frequency, so studying something more might signal to your brain that you're going to see it again. Like if you see someone 3 times, it might be important to remember them, right? So you might reactivate them, but if you only see them once, maybe not. Also, if you see someone 3 times in different contexts, they might become more important to activate that person versus the contextual information. So those are the kinds of questions we want to ask, like what's adaptive? What is the brain listening to? And we're just having fun kind of inverting the question instead of being top down in this moment. We want to be like data driven, and that's why we came upon this uncertainty. So maybe experiences that you haven't quite figured out yet. There's an effect in psychology called the Zeigarnik effect, where we tend to bring back to mind things that are unfinished. So if you forgot to put your like laundry in the dryer this morning, it comes back to mind later in the day, right? So that is that a moment of replay? Is that the brain kind of continuing to like remind you literally of things that are not finished yet? And once something becomes finished, like you're really done with it, does it undergo suppression of replay? Does the brain like put it aside for a while because it moves on to something else? Those are all the questions that we want to start to ask in the lab.
I guess I'll ask a question if you really twist my arm. I think you sort of answered this, but I'm intrigued by the difference between something like rehearsal, rumination, et cetera, which I think of as a very conscious, deliberate process, even though it can come to us unbidden, and the kind of like, biologically instantiated replay that we see in the brain. And do you think that the mechanisms of those are similar? And I guess a related question is a quiet mind, like the mind of somebody lying in the scanner, post-encoding, a more efficient replayer, or what are your thoughts on that? Yeah.
Yeah, I like that question. Well, I'm going to answer that one first.
Yeah,
and then
go
you'll
for
have
it.
to remind me of the first one. Okay. We got that question a lot in the beginning. We did many studies where we had people encoding and then resting with eyes closed. And our very first paper was published in when it was published in Neuron, it got a lot of press, and the Italians loved it. Their headlines were all like, take a break, have an espresso, and they really loved that, like, their lifestyle was like really conducive to like enhanced cognition. So we started to think about, like, what counts as a break? And I just talked about walking through a door might be enough, right? So we have done studies where instead of moving from an encoding task of paired associates into rest, we had people do math. So we have them in the scanner answering math questions. So it's a very different brain state. It activates different brain regions, and we're still able to see that hippocampal cortical activity during math predicts memory. So we didn't tell the Italians about that answer, but it turns out, so we don't know the extent of that, though. We don't know, are there certain high effort, attentional, demanding states that are inhibiting for replay? I don't know the answer to that. We've only done a little bit of work to show that you can still measure it in when people are doing another task. And I think that makes sense for how we conduct our days. Like, if you can't stop for 5 minutes and just close your eyes, which I recommend doing. But if you can't, it doesn't mean that the brain isn't taking advantage of the shift in context to be able to reactivate. But it's still an open question.
Well, I am Italian, and I do like the espresso break idea.
I said that for you, yeah.
Are there other questions? Because my second question we can save for our dinner chatter,
Okay.
but are there other audience questions? Oh, I see. Okay, so thank you, everybody. I think we're going to wrap up now. And that was a really interesting conversation that we all had, and your work is just so wonderful and so fascinating and innovative. Thank you so much. Before we scoot out of here, I just want to remind everybody that there's a reception downstairs, so please join us in celebrating Lila. Thank you. Thank you all.